So now our next topic we are looking for synchronous growth and the further it is batch or continuous culture. What is the first point? Here we are going to understand the three or the two phenomena. It is synchronous. Can you have any things there? What is meant by synchronous growth or culture? The word synchronous, or you may know the word sync. At the same time, syncing. You people are using the mobiles? Yeah. So maybe our mobile, right? That is like a context. It generally keeps synced with our email ID. So whatever you are going to add, okay, any numbers you have added, it is going to be uploaded or going to connect it to our Gmail or whatever the email ID we have kept. So it is synced with one to another. So one at a time, what you are going to do, it is going to get connected with another. So it is called the syncing. If group is we are looking as the time change, one has to change their attire. Suppose it was like a Ganesh Utsav before a few days. So everyone is allowed to come in some traditional dress. Some may come, some may not. So it is syncing with, you are synced with the phenomena. Here it is like the syncing means, just I'm giving the example of like we people are using generally the mobiles, whatever it is, Android or any else. As you add any context into it, it is going to get uploaded into your connected Gmail or other IDs. So one at a time, what is happening, it is going to connect with another. So here if you are looking for the synchronous growth or the culture that is related to the bacteria. So we are looking for, what generally happens, synchronous growth, it is going to show in some different pattern. That which time, log number of the cell, if we are looking for the leg phase or the log phase, right? So in general, what happens in growth curve, it is like curved like structure is forming. But in synchronous, all the things must go in a similar pattern. As the time is going, the products may be generated or the things may happen. It should or must sync. The cell size increase and the cell division must go with one to another parallel. Parallel thing is required to go. So it is like maybe somewhat limiting. In leg to log phase, what happened? All are going into cell division most probably. In stationary phase, cell dying and they are getting mature. But in synchronous growth, both the things should go together. So some of the cells may go for cell division, one another cells are going for the maturity. So there is we are looking for. So how we can attain such things? By adding or limiting a growth of the bacteria. If I'm looking that I may call and tell you that you are going to go in a lab. Okay. You have the so many choices to reach the lab. You may have the other stations. If I told maybe at right now that we are going to meet at 10:40. So you are having the so many things, so many aspects may be completed. Maybe a lecture here and there. Or maybe I just announced at 10:30, we are going to meet at 10:40. Even in that 10 minutes period, from here to reach to the lab, you may have the certain options. You may go to washroom for a canteen, or some person is already calling some since long. You have to give the answers, right? So many aspects may be connected with one to another. But if all the class member, it is been directed and all are looking and syncing with the given time period. Okay, so it is called the syncing. Though you have time period, but you are not moving here and there. Okay, it may be sometimes the exams are there, right? In that conditions, whatever the condition is there, you are going to get the permissions only then you are going for any washroom or in a water room or the canteen. So one message only at 10:30 it is given to reach at 10:40. So you are compliant. So what I have done, normal routine time, there is a no problem. You can move. But I have limited during that time period because it's the exam session time is there. You people by yourself, you are limiting. If it is not get limited, you are going to limited by the other faculties. Okay, otherwise you are ready to give the answer. So you have to manage this pattern. Maybe it is minor leg or log, almost it's going in parallel. Maybe one or students may ask for the permissions, may I go for the water room or the washroom, or maybe a certain times the canteen. So from the morning, your mobile was switched off. One has to. If you are going to caught with the mobile, you are ready for the further consequences, right? Even that is other material. Sometimes we people are used to it nowadays. That birds, right? Keeping. Yesterday, some exams were going on. Some students has kept. Right now, it's like we are in a pocket, and here we are keeping. Right? He was not using, but he's been caught by the supervisor from the outer side staying with it. So if nobody can say and cannot deny at that time that you are not having that gazettes because it's not permitted. I don't know. Maybe that outer side, it is maybe connected. When you have used, when you have kept, nobody knows. But you were caught with it. So ready for the consequences like this. So then you will be ready, going to check twice by chance. Maybe it is like a pen drive. We know that the pen drive can be operated with certain instrument only. Either you can open or use in the your mobile, or it can be operated with iPod and others. Earlier people were using just a memory card chips. So how I may know? Pen drive itself via Bluetooth or another way manner going to get connected with the birds and you may able to listen. So many things are there. So you will be then after going to limited. So here we are looking for the bacterial growth. So they will get limited. So all the organisms to achieve, we are not looking for the doing the certain experiment right now, but chemical or physical factor, we are going to manage in a way that all the microbes will grow in one steady common graph. They are not looking for the leg log phase. Steady increase in their cell number is going to happen. That's called synchronous culture or synchronous growth. So they sync. That means they are going to sync with the what it is being given as the instruction. Here the instruction is the physical or the chemical factors. If you limit them with this, they have to follow. If they are not going to follow it, they are going to get rejected. Okay, so this is the very basic phenomena people are using into it. Here also we can measure using the synchronous cell division with this continuous steady number of the cells are increasing. In case of other asynchronous culture, that initially leg or log phase is there, cell division is higher and higher and higher. Okay, maybe on stationary phase it may limit it. So phase wise is changed, but here it is proper. Every cell is doubling one at a time. If we have kept for the doubling itself, or if we have kept for going into the very limited, so they are maybe parallel to the stationary phase. So they are not going to get for more in a cell division. So this is called the limitations over it. So cell division in synchronous growth is continuous either, or it may be limited with time period. So we may increase the temperature or may decrease the temperatures or may add some chemicals they can control. Okay, so this is all synchronous culture. Next one is a continuous culture. Just I can show the pictures here. Generally, it's better I think to talk first batch culture. Yeah, this one is the regular thing. In a flask we are maintaining, or same can be maintained into fermenter. Large, small vessel or the large vessels, fermenter if we are putting. So whatever in a first batch culture, what we are doing, or it is in maybe in a smaller flask, we may inoculate a medium it having every sort of things required for the growth of bacteria. You may keep for the 24 hours, 48 or 72, or as made low, it goes on. Some products are going to synthesize in fermentations after a 1 week, depend upon the products. So it is just one at a time. Here we are just taking the flask. Some of the times we may add something. If it is a larger, have you learned about the fermentation technology? Yes, I think bioprocess technology one paper is there, maybe in semester 5 it's coming, I'm not sure. So generally in fermenter, agitator, aerators are there. Do you know about it? Aeration, agitation. Just let me show just few things, not more. You are going to learn otherwise maybe later. So it is like, maybe it is just I'm showing in a larger level. Have you seen, yes, our washing machine? Everyone maybe have seen or may have there at home. At the base, something is rotating. Maybe it is left, right, or something is going on. So it is nothing but a or agitator. It's called agitator. So it agitates. Sometimes people, when you are standing here or sleeping, okay, in a bus or sitting here together, some people are continuously waves their legs. Okay, it is their normal, left, right, or something and something going on, right? They may be not knowing what's happening and going on, right? But it is called the agitating. Okay, those who are volatile in the nature, sometimes by chance, everyone has, or it is not always, or sometimes you are listening some music, you are in your voice, and it may happen. So it is nothing but the agitator. Maybe soft. This one is called the soft. Maybe it is attached to the lid, or sometimes it is from the below. It is like we are having the fan. Okay, fan can be here. It is required to hang it. Same can be on a wall mount possible, or a separate unit also there. So for getting it, so it is called agitator. Agitators are being supplied either in a shaft or into the agitator with a bubbles forming unit. So from there, you can send the pure air. It is like that. Maybe you have seen the aquarium at some other place. Nanu machli ghar jo hoy na, when you are going in hospitals or into good restaurants. So continuously bubbles are coming. That's called the aeration. Okay. So why it is required aerations? For the feces, oxygen is required. For here, it is our bacteria. So continuous, this agitator is moving either in one direction or in revert. The rate may change. That is the RPM, revolution per minute, may change as per the requirement. If you are going to rotate very fast, bacteria is going to die. It is like that. You are sitting here. If I'm in an invigilator, in your examinations may be going on. Paper is given. After every 10 minutes, I may ask everyone to sit here and there. Initially, you may not going to mind. But every 10 minutes, I'm going to call anyone and going to ask to say, sit here and there. What's going to happen? Will you like it? Why? It is called the agitations. Okay. But as I am a supervisor, I'm having that power to do. Okay. I'm having the power to do, to take you here and there, any of the damn place. Okay. Then I may keep the switch on, switch off fans, anything. So we are playing with. I'm going to play with you for a while. If I'm going to look to agitate you people. Like this only. Here we are doing. If you are going to put the agitator higher speed, microbes may like. Okay, if you are at a random and a canteen or some other place are there, you will, on your leisure time, you will like to move here and there. Or I have taken you in the playground. You will never mind to move here and there. I may say, you people 4 groups sit here, there. You will say, okay, sir, it is 1 or 2 hours. We are coming here for the playing and others. You will never mind. But your exams, whether you are knowing it or nothing, 70 marks paper, you hardly know about the 20 marks something. Sometimes happens. Okay, some topic is there, but you won't like to get move here and there. Like this only bacteria also. So we have to limit the speed and others. Even the aeration. If it is right now, it's a cool temperature. Nothing is a problematic. Same it is in winter, in a February, about the temperature is a 10 to 12 degrees outside. Everyone has weird, including the supervisor with caps, right? Having the jackets, and I am going to switch on the fan. I'm going to give the erection. It's my purpose to give the erection to you. What you like? Would you like it? Or it is in a summer, hot summer conditions. I may say I must going to switch off the fan because the voice is coming. I'm not able to concentrate and check what you are doing. You cannot say we are not because I'm a supervisor over that time period. Or some supervisor is coming from the outside. Just I'm giving my name. You cannot do anything like this. So bacteria, as you consider yourself as a bacteria, in certain temperature environment, you will like certain conditions somehow. So we have to control or to optimize the phenomena. Why I'm telling this thing? To optimize. Everyone is like to live in some optimized condition. Okay, optimize means in a proper in their environment. There they can flourish in their life. So here it is like a leg to leg phase. Bacteria are looking to leg to stationary phase. Like this only, if you are coming for an hour, you can sit. Okay, in a class. After 1 hour, if I'm going to continue for the second lectures, everyone is going to ask initially for permissions. Okay, if I have denied, half an hour is going on, will start. As I'm moving, okay, it's going to happen because you have likely or dislikely or not likely. It is going to be controlled in another way. So you may start to do good as well as some mischievous things. One has to do, or everyone is not going to do. But by chance, supervisor or someone else, the class teacher, may likely to continue even that conditions. You may sit up to 2 hours. Then it will be again maximum 5 minutes break is given. Same person is coming for the continues for 2 hours. What's going to happen? Will you like it? You may say yes, but from the inner side you want. Okay, so like this only, or all the physiology is concerned with this. So we are looking for two major things. Why I'm telling this thing? That in a batch culture, we should provide the optimized condition for the growth of bacteria. If I want that in a you are coming in the papers for writing the paper, I'm your invigilator. Sometimes some having the some frighten up, I may calm down, go for the washroom, take the water bottles, no problem. You won't be enough. Other persons also get smooth and calm. Okay, the environment is going to lessen down. Everyone is writing properly. Same, and the write-up also going to be a better. But same in that conditions, I may start and come to shout many things here and there. At a 5:40, no one is going to move here and there. Please sit there. Everyone is sitting. Okay, so one is agitating. So it is become difficult. Now, in second condition, this is related to batch culture. It is what about the continuous culture? In continuous culture, what we are looking, sometimes we are restricting when the continuous culture is far better, that either or we are looking for number of the cell to be grown. Okay, we are just, maybe it's a yeast culture. Yeast cells are there. We are looking for the Becker cyst. So all the cell must be remain in the leg to log phase. Or I'm looking where that bacteria are certain products they are synthesizing. So they must be in a one another afterwards stationary phase. So what I'm going to do, one at a time, whenever I'm going to do add the sterile cell others, overflow is there. I'm going to take the material outer side. I'm adding the material inside, and after I'm going to take out the something from the another place. So as the from the another phase, either it's a bacteria or the another material, that is our bacterial suspension, or maybe secondary metabolites are being synthesized. It's like a citric acid. Citric acid is synthesized. I'm going to take it out. I will purify and going to bottle it out and sell it. Okay, it is maybe like we may keep a filter. So from the filter, all the microbes are going to not move out of it. So now the broth is there. Within the broth, I may consider that citric acid is there. So citric acid, I'm going to make either their tablets or liquid is given. So generally what happened when you are taking like 500 mg tablets and others, okay, like in paracetamol, what you are taking, like it is written as 500 mg, but when you are going to weigh the tablets, it may be more than that. So additional things are always there. So while preparing, another media constituents are going to be there, but it's not going to harm us. Important constituent only is our tablets containing that medicines. So we are not looking for others, but we are looking for this. So continuous culture are being produced or prepared for to keep the bacteria either into log or in a stationary phase. So it's called the continuous culture. It may goes for maybe sometimes for a month. If you are in pharma companies, 1 month to 6 months, continuous process is going on. Yes, we have to monitor from the outer side. This part is required to monitor from the outer side also, like their pH, temperature. It is like a 24 hours is running. So people are working in the 3 shifts. It's required to recruit our staff in a 3 shift continuous. So all are coming and going and coming and coming. So like that. So bacteria not knowing that I have to be there. So we have to check the inflow and outflow, and the continuous culture is managed. So this is the some basic phenomena related to it. So batch versus continuous culture, where to use the someone, like it is sir or it's written. Okay. So when you are looking for the product is what is important. Okay. So people may either go for the batch culture or a continuous culture. Same vessel can be used for a batch culture or also in a continuous culture. So depend upon our requirements. One may go for the batch or at a higher level of a continuous culture. Right. So I think it was the last slide in which, yeah, that's how is in a different format of graph. So time versus concentration. In batch culture, substrate what we have added, it's going to decrease, product is going to synthesize continuously. While in case of a continuous culture, substrate and the product concentration is limited and going together. That we are going to add continuously the substrate, the product formation is going to be sync with it. Okay, both things are going to get together in that format. Here it is get depleted. So if you have taken like simple the E. coli growth you have taken, and you are going to keep for the incubations. Maybe after 4 or 8 days or 10 days, everything is get utilized. Microbes may be there, but they are in a, then after in that space. That means everything is converted into product. Here might be a product is a cell number. In our case, every day we are doing batch culture. That is a inoculum is given. That is a batch culture related to cell number. Product is our cell number. Or if we are looking for, they are as a secondary metabolite. Sometimes the other organism, if they have grown, so they are start to release certain secondary metabolites. The cell number is increasing, but afterwards limited. And in stationary phase, they may start to produce certain products. But it also going to limit it after one time period. So maybe this product formations and the substrate, their timeline may change. Sometimes that is a cell when move into stationary phase, then only our product formations may start. Substrate utilizations versus product. While in case of here in a continuous culture, what's going to happen? It's going to add additions and deletions are going together. So always we are going to add substrate continuously and taking the product outer side also. So I hope this point may be clear to everyone. Okay. So let me move to the next point. Already we have performed this topic, I think. Now we are going to learn about it. That is physical factor required for microbial growth. Already we have performed that in experimental part. Okay, already we have done. Though I have discussed many a times, so we are going to discuss now more in more detail. So what are the physical factors according to in this part? Like temperature is a pH, osmotic pressure, hydrostatic pressure, radiations. So many factors we may go. So we will take one by one. Temperatures. Okay, what's going to happen? Optimal growth temperature, maximum growth parameter temperature, minimal growth temperature. So as you limit, it is optimized. We are going to use this word if you are working in microbiology lifetime. Every organisms required to work in their optimized condition only. So it is there. So minimum growth, that is the lowest temperature where minimal number of the growth may be there. We have still time, no problem. So we will continue in that minimal growth temperature. So as you decrease, maybe it's a 37 for certain bacteria. So till how much you can decrease that microbes may continue to grow. So for that, we need to keep our tubes into different temperatures. If I'm going to keep it at 0, there is a no growth. Otherwise, I may keep it at 25, there may be certain growth. Okay, so they are optimum at 37. It is like that. We people generally having our temperature according to our climates where we are born or brought up. But by chance, if we are going into like mountain, Himalaya, or into foreign countries in the lower temperature. Okay, it may be during the winter if you are going. Okay, in a Sydney or other places are there, the temperature may be at -20. You will feel yourself here in deep freeze. You can survive. You are not going to die. But yes, you have to wear the certain things. By chance, if you forget and if you start to move outside, on a next hour, everything may change because you are not adapted. So it is our minimal growth temperature. You can survive, but if you are taken, it's -20, it's okay. If there the temperature is -40, crystallizations start to get formed. You are in Antarctica. Just imagine that. So there is a major problem. Then the optimal growth time range or timeline or temperature range line 20 to 40. There will be no much variations or no harm. Okay. Then the maximum growth temperature. Okay, the highest temperature which growth occurs only slightly above the point of frequent decrease the microbes by inactivating critical enzymes. It is like that if you are in Sahara or the other place. Temperature more than 50 degrees. For us, it is 50 we can tolerate. Afterward, our every sort of the mind and the balance, physiological things are going to get disturbed as the temperature increase. Like generally we are knowing that for body temperature, we are managing in the Fahrenheit. 102, 105 Fahrenheit temperatures may go. Body temperature, that is 37 to 38 to 40 degrees centigrade. Internal temperature may go. Then the drastic change happen. We have to keep the eyes on our mind. Why? It is a vital organ. We are going to manage first our vital organ. Other organ system are getting disturbed. But we are going to first save our vital organ. So maybe the ice packs and everything we start to put on our heads. So it is the requirement. Otherwise, it may going to disturb other places. So how the temperature has increased? That is in immunology, maybe you are looking the adaptive and the acquired immunity, maybe you have learned or going to learn maybe future. So our body has started to increase the temperature. How the temperature increase? That is the metabolic rate may change. That is like our powerhouse called the mitochondria. Okay, it starts to increase the temperature. So what we are going to do, we limit it. So at the cellular level, we are limiting them. Okay, we are going to limit that. You need not to activate that enzyme, need not to go for the increase the temperature. It's like thermodynamics. Our body, the temperature has increased. So thermodynamically, we need to balance. So pharmacology, pharmacognosy, and pharmacokinetics. That is a part of the pharmacy related work is there. So body is increasing the temperature. We already know. Sometimes someone is sitting, just please check, I'm not feeling well. Oh, yes, having fever, take rest. Okay. Sometimes we teachers also say, no worry, rest well, don't talk with one to another. Otherwise, by ourselves we decrease. You don't like to eat even whatever the food product is given. Why? Or test, that is in test bud, the receptor on a tongue only in optimized temperature and conditions active well, not in every condition. Temperature, cellular level has increased. So we are limiting with paracetamol and others. So at cellular level, they are going and change the temperature. My enzymatic action is disturbed, it's going to get imbalanced. Then this sort of the things happen. Even it's sometimes psychological change also disturbing. It is like that. Whatever you are eating, something you may like or not like. But at that time, some call has came. Your friends or someone has got some bumper price. You will in jolly mood even. Or you has got something. Right. And afterwards, maybe at yes, you are going to get free, you are going to get something special. Or it's like a gift, something is there. You are in jolly mood. Same at a time. You receive the some call for some very bad news. Okay, someone has got your nearby, got accident or something. Even you are eating any good things, you want like to do chew even. That taste bud and linking with our neurological part, it get disturbed. The other hormones and other has may have secreted during this time, the process is disturbed. Even you are eating your rasgulla, maybe like, or a pizza you are eating, you want like to eat more because something has happened, your eyes are tearing. How it's happened? That hormones has started to release at the one to another place. The whole body mechanism is get disturbed. Simple one thing. The reason is what? Something has happened. And all the your processes disturbed. So metabolic change. So here these all are connected. Physiological parameters, maybe it is internal or external factors. Either the simple the temperature has increased, you may feel chill or you may feel the feveric condition. Right. Or sometimes that call has came, everyone is good, but the same only one person is getting problem. Okay. So some factor, it is called the psychological factor. Let's not connect with the bacteria. We are going to say that your mom died. They are never mind. They don't know about their mom, right? Because they are looking in the which fission? Binary fission is going on. So mother cell became the daughter cell. So they don't know about their mother. Who is mother? Who is my mother? Okay, they always became independent. So they are not worrying. But why we are worrying? We are connected and one individual is going to develop another one. So everyone is individual but connected with one to another with certain relationship. And that's why the problem may start. Otherwise, not. So there is some different is the physiological, two additional psychological parameter is coming. So here is a optimum maximum temperature, growth rate and the temperature. Huh? Yeah, that's what I'm showing. Optimum means the temperature is good, growth rate, bacteria will grow properly. That is their like environment. Optimum means their like thing. Okay. It is continuously, if I'm going to come here for a 10-15 minutes, I may give the sandwich and others. You always like my lecture. But on the second, I may just start to call, I may going to call your parents and so and so. Your shift to make in this, you will not like my lecture. Simple phenomena. What happened? Just I have changed my mood. Your mood is changed. You get disturbed. If I'm in jolly mode, sometimes telling like these people, like papers are checking or going on. Sir, cha nahi pidi hoy. Sir ne cha piyudo hoy એટલે આપણું કામ થઈ જાય. Right? If I'm giving the 10 minutes or 15 minutes more break, and I'm asking for the practical, you will be ready. But if I'm continuously, you may not like. Right? So these are the factors are there. So what is the optimum conditions proper? So temperature. So we are playing with the bacteria. So for every microbes, it may change. So we are going to talk in later that part, psychrophiles. Now the classification of the bacteria according to the temperature. Psychrophiles growing at a 0 degree centigrade. They are very happy at a lower temperature. So especially if you are going into like Himalaya and other places, there also microbial communities growing. Very low temperature at the deep phase. Maybe their growth phase may be lower. Okay, there, like here we have seen about the 20 minutes. It may be a 240 minutes or maybe a 2400 minutes, maybe for one to another cell division. The psychrophiles are following in that category. Other group can grow. The optimal growth temperature may be about 15 degrees. 0 to 15 in a range may they grow. Okay. The other groups, they can grow at a 0 at the higher of temperature, usually 20 to 30, not above to it. So psychrophile, thermophile, and mesophile. We can classify all the microbes maybe into this category. Okay. The psychrophiles, lower temperature, mesophiles in optimum temperature is maybe you can say 20 to 40 in that. Above 40, if you are going, that is called the thermophiles. We are also following in this category, mesophiles. We are mesophilic animals, and other are psychrophiles. We need to keep our temperature warm from the inside. You can go in a mountain, it is a minus 20 even, but you have to manage yourself. Your body temperature is going to fall down below a certain temperature, you won't survive. Or even if it is increased more than that, you won't survive. It is the temperature range. So psychrophile, we can temperature at 0 to 30. Here, yes, here may range change from here. Common optimum 20. They may be written as temperature at 15 to 40, and 35 is the optimum. Thermophile 45 to 80, optimum 60. So you can sometimes may think that there is a range are crossing with one to another. That means it is not like that. That 15 is there. So below to 15, they cannot survive. Their limitations timeline is there. Below to 15, they cannot. This is their optimum range. So you can also manage yourself till 15. 15 to 30 to 40. If going increase, we may need to either start the fan or the cool AC is required. Otherwise, the heater is required to survive. So we are managing in that way. So range changes into one to another place. So we may classify our microbes based on their temperature. Yes, these questions may be like physical factors, as well as I'm going to discuss about the chemical factors. All can be asked in the 8-mark question. Or individual can be asked into 2 or 6 or 4, 6, 8 marks pattern, like just related to the physical factor temperature. Okay, then you have to give these classifications into it. Okay. So temperature optima. Okay, so cold living. Sometimes we are using the word optimizations, optimum or the optima. They are, you can say the adjective and adverb form one to another or not. Okay, so optima, temperature optima, it's called the phenomena. To manage it, or optimizations, it's a procedure to manage it, or we are going to learn about it. And optimum, that is the one is suitable temperature for that organism. So in optimizations, we are going to know that what is the optimum temperature for their growth. And to understand this phenomena, we are going to call as a temperature optima. That optimizations to be done to check the optimum temperature of that given organism. So don't get confused within the 3 words. Optimum, optimizations, and optima. Everything can be asked in a 1-line question. What is optimizations? What is optimum temperature? Or what is temperature optima? So don't get confused within it. Optimizations or the optimum temperature. First start with the optimum temperature. Or this like E. coli, just keep in example 37 degrees. If I'm going to give some unknown bacterial solutions to you, I know it is E. coli, but you maybe not know it. So what you can do? You may keep certain tubes or on a certain different degree temperature, and you are going to plot the graph. Then you will say that this temperature is 37 degrees there, where my bacterial sample, what you have given, is growing well. So you are optimizing it to find it out optimum temperature, and this phenomenon is called the temperature optimum. Okay. So temperature optima, yes. What happened during below or higher range if we are going? First problem, it start with their enzymatic disturbance, metabolic instability. Another, there is we are calling as a structural and functional protein. Do you know about it? Structural and functional? Structural protein and functional protein. The difference between two. Okay, then functional. Like, yeah, right. So structural and functional. Structural is a component of the cell. Maybe it's on a membrane. Organelles are there. Organelles also with their protein, or you can having the cytoskeletal material is there. So everyone is maybe connected with. So it's called the structural. Functional, that is related to the enzymatic process. Okay, it may be as a part of the channels. Maybe you are learning the transport system. So it is also a functional part. The structural and functional over it. So enzymatic reactions we are learning. So either of, mainly it is maybe disturbing their structural or the functional protein first. If the temperature may increase, we know that with increase in temperature, first most important part is that water will start to lose. As the water scarcity may happen, osmosis may start. As we are going to get sweat with increase in temperature, right? Sweating is going to happen, your water is going to get dropped down. It is like that. Sometimes we are going in some trekking. We are having the one just water bottle. You have completed in just 1 hour. And trekking is required to do for at least 8 hours long more. Initially, you are running very well. Then your intake for the water is now totally completed. And exhaust, with exhaustions, with inhalations, and with sweating, your water you are losing. One time will come, then your activity may also reduce. Right. So like this only, with temperature, first important phenomena related to the water, it may start. Another is the temperature may increase. Our enzymes are working at their own optimum temperature. Their metabolic rate may decrease. Other is like a lipid and carbohydrate. Their structure may destabilize. If anything you are going to keep in the hot palm, okay, something is going to get disturbed. So like that also, others, all the cellular components are going to get disturbed. And this may lead to halt of the bacteria or the populations, or they may die afterwards. So here the psychrophile, mesophile, and thermophile, there one comparative graph is given. So they may ready to grow, may survive at -15, better at 0 to 5 degrees centigrade. Then their growth may sink or reduce down. Another is like a optimum for another group of bacteria, mesophiles, green color. So they may start to grow, they may survive. They are very good at 35 to 37. Then their growth may reduce 45 to 50. Thermophiles are moving in another direction. So here you can see rate of growth. Almost the higher range is coming for the mesophile. Here they are growing, but the growth rate with time is changing. So growth rate or in mesophile is very high compared to another. What could be the reason for it? That if you are looking, I have shown the 3 graph here, right? Here almost psychro and thermophiles are at one parallel, but you can see the optimum given and the height of that graph is higher in case of mesophile. Why it is? Yes, it is optimum. This one is optimum for this bacteria. This one is the optimum temperature for this bacterial group. And here it is. But even after, their growth rate is lower than the mesophile. That is optimum. That's what I'm telling. Yeah, pardon? How long? Yeah, why they are limiting? If it is like that, those who are living in the very low temperature, Antarctic, or in certain countries where the low temperature is very low. Certain countries, the temperature is maybe in this condition, temperaments. Other is a very high, Sahara and others. Just you may distribute into 3. where highest amount of best proper activity, even for the other bacteria, if you are looking for the other vegetations also, or the diversity is observed in this temperament only. Why? Why the mother nature is supporting one particular temperature very best? Getting my question? समझाई છે પ્રશ્ન મારો? Enzymes, but here also they do have their own enzyme. Otherwise, they won't survive. Yes, that's why I agree. Here also they are best working in this. Sometimes I may give you another way example. Some people are reading for reading phenomena. Right now, the major populations are sometimes early bird. Wake up at the morning during exams, not daily. Okay, most probably they may wake up very early, or during a 10+2 may have that habit, early reading. It's going on. Sometimes people are the nocturnal. At night, 2 o'clock, they are reading. What there are other activities going on, whatever. But I'm telling that. So it is the ability or capability. But they cannot at 11 or 12 o'clock they slip down. Okay, to stay up to 2 o'clock, it's also whether you are reading or not, but you are able to wake up yourself and you can sustain that time period. Either of the activities up to 2 o'clock, maybe then after it may reduce. Why? Why it reduce after 2 o'clock? Generally, maybe at 2 o'clock, 1 to 2, up till maximum 3 o'clock, right? Then after also nocturnal guys going to get slip down. Why? Or those who are early bird, maximum they may wake up at a 4, not at 3, maybe at 4:30 or 5 o'clock, not before. What could be the reason? Maybe it's for the reading, maybe for the other aspects, but then that activity is going to get reduced down, right? And it's also connected with the age factors, as in lag to log phase, stationary phase, where it is bacteria, our age also is sync with it, right? But then after certain activities may reduce down. Like this here, I'm asking that optimum for one to another is changing from one to another. And there is a higher growth rate only is observed in mesophyte. Why Mother Nature has kept this? Right now, in an earlier phase, if I'm just going to like a survey, most probably going to say that we may slip down at a 12 or 12:30 or 2 o'clock. Maximum of you may agree, right? Earlier, it may be a revert. They are early birds. Now, the change in scenario for all the people, not just you. Okay, maybe your younger guys or your elders are having such phenomena. Either we may attach to the TV or the other gadgets or other thing is going on, right? But all the phenomena, entire world is moving in different formats. The earlier format was different, now it's changing. So their optimization has changed. And accordingly, their growth rate is also changing. But here it is this. So all our environment of our globally, if you are looking, right, it may range and put at 1 degree particular average temperature. So it is like thinking all organism try to adapt in that place. All the environment, all the things has adapted to one given temperature in this line, 30 to 40. If you are looking for the average temperature of our global environment, whether it is you are in the north or the east or everywhere, globally, if you're looking for certain temperature, it's ranging in this. So all the organism, this also looking to shift, but they cannot because they have already adapted. It is like that. Those who are getting the punishment, now they are in prison. They already caught. They have done certain bad things. They caught. They want to come and regular life. They want to after the imprisonment also they are coming outside. Okay, the bylaws what is told? Once their punishment is done, person has right to live as a normal. but our society is looking them as a different always. So they have already moved. They cannot bear. Like this only, our temperature, they already moved into higher to lower, but their rate is limited because our whole world is moving in one particular direction. Okay, if I'm going to take you in different format, I know that all people are going to sleep down generally at 12, around 12. Okay, but I may take you in certain trips or something and push you everyone to sleep down at 10 o'clock and you have to wake up at 4 o'clock for a 1 month. You people will like, may not. Schedule may be in that format only. You need to compulsorily adapt with it. Otherwise, not. But I may know these things. I may keep the certain things to have the playing, some games, and everything is going at 12 o'clock year around. Those maybe like to sit, they may sit, play the music, or games are going on. They may like to it. We are going to get wake up, and at 9 o'clock, our trip will start. Everyone will enjoy because it is sync with the whole world is going on. This phenomena, so you will happy and the growth will be higher. If I'm taking in the different odd sector, growth will be there. Likely or dislikely, growth will be there, but it may not that much. And that is why our rate of the growth in both is changing. Okay, then the mesophiles, they are growing well at 20 to 40 degrees centigrade. The same thing is written related to thermophiles. They are in 50. What makes them to survive at after the 50 degrees centigrade? Or what they something make them to survive below to 20 degree or in the lower temperature? What may be a change? It is maybe there. Can you just think? Yes, simple first example I may say that I have told mesophilic. If I'm going to increase their temperature, functional protein or the structural protein is going to get disturbed, right? So they have gone for certain adaptation. It's called the like chaperones. Have you heard about the chaperones or chaperones? Okay. Or headstock protein? No worry. Maybe our maybe afterwards you are going to learn. Just I'm giving the few examples that parts that body is going to synthesize, that bacterial cells are going to synthesize certain molecules within them, so they won't going to die at higher temperature. First phenomena is what? Temperature increase, osmosis happens, right? So what is the problem going to happen in osmosis? Water is moved outside. Okay, salt concentration is very high in outer, here it is low. That is a normal phenomena. So it may take some osmotic balancer within it. So they may increase the osmotic pressures from the beginning. Generally, or the some acidophiles. When you are going near to, you can say, lava, where is the volcanoes are, eruption is there. So one is the temperature rise and acidic condition. So thermophiles are generally the acidophiles also. So within that, so they will start to accumulate certain thing within their cell. How we may survive? We cannot include anything within our body. So we may warm up outer side. Okay, we may cover ourselves using certain clothes. Yes, other things we may do, like if those who are in army and others, they may start to drink certain alcohols and others. Why it is required? To maintain their body temperature. If they want, the temperature balance will disturb. So like that, we may start to consume certain things to balance it. Like this, our bacteria is doing. They start to get the things from the environment and grab inside. Then only they will survive. Otherwise, not. Okay, that is related to lower temperature. But it is in higher temperature. If you start to drink a lot and the temperature is very high, you will start to burn from inside. Acidity and everything will start to happen. If you start to eat very high veg to non-veg or the chilly conditions, and you are in the summer, you will in a problem and trouble. So according to the temperature, every organism has adapted certain points. That adaptations, they make them either in psychrophilic or in a thermophilic conditions. All were in the initially in mesophilic conditions, but according to their geographical locations, they have done some adaptation, and these adaptations has led to development of certain chemicals over their outer layer. They may have the additional membrane like structure within them to survive into different conditions or in temperature, or they may have developed certain enzymatic process in a way that to survive in that place. So certain heat-suck protein, it's called, or the chaperones, for a lower as well as for the higher temperature, they able to survive in that condition. We don't have the heat-suck proteins, but the other microbes do have. Okay, so they able to manage in that condition. So this chart is showing once again the same thing. Yes, just one point, then I will make you free. I can understand also. Called the hyperthermophiles above the 80 degrees centigrade. That is the hyperthermophiles or the hypercycrophiles. Here it is shown the cycrotolerant cycrophiles, extreme cycrophiles. They can survive at a very lower temperature even, or the very high temperature. So we are going to call them as the extreme cycrophiles or the extreme thermophiles. So they can manage themselves at a very high temperature as well as very low temperature. So that is why the people are doing certain experimentations. When they are going in a space, they may bring certain bacterial suspension and others, fungi or the algae, and they are looking to study over there. They may develop their studies over it. Whether bacteria start to adapt into different format first, that is mesophile may convert it into thermophiles or the extremothermophiles or what. So according to their adaptations, we can learn that how we may be able to adapt or the other organisms can adapt in this environment, or what are the effects of their temperature and their gravity on the life of that organisms is going to happen. Accordingly, we may develop certain models. You may know the neural networks, neural net in bioinformatics. Okay, that may be coming in the next semester 5. Okay, that is the neural networks are there. That is what we are looking. That is the neural logical parts. Same like that, some bioinformatics tools are coming. So we may, one model is there, another is developed. So I'm not looking to move ahead in these directions. We will continue for the PH maybe next class. Let's just make it put. Just put your attendance, no problem. We can continue for some questions. What application can do with like PCR, polymerase chain reaction. Maybe you are going to perform in molecular biology, maybe later. Okay. So when you want to work or the certain experimental part is required, so we can take the help of it. The microbes are growing at a very high temperature. So they having their enzymatic setup. So DNA replications, maybe you are knowing. Okay, so who doing the replications? DNA tech polymerase. Sorry, DNA polymerase is doing it. So one organism very well known is called the Thermus aquaticus. It is growing at a near about 100 degrees centigrade. So DNA polymerase is taken from it. So it can survive at a higher temperature also. So in a machine, PCR machine, our temperature is required to increase. Maybe it's a 90 to 100 degrees centigrade. So this is not going to get deactivated. So according to our requirement, yes, in dairy technology, what happens? We are using the ice cream to be get chilled. Maybe you are using, maybe you have learned preparing the ice cream at home. Yes, that's good. What we are adding? Custard powder. Powder we are adding or not? Correct, બનાવું? Nah, correct, બનાવજો. Correct. Some powders are coming that you are adding. Okay, so it is a mixture of certain proteins. Huh? So that we are just adding from, maybe it's a plant product. But those who are working or ice creams and from the Hemos or so many companies are coming, what they are adding? The protein is taken out from the cycrophiles. So it can retain the temperature at lower. Okay, if you are going to keep outside, some companies, if you are going to take it melts very faster. Amol and other. Oh, just we are talking and within the time your ice cream get melted down. But from the others, it may take about 15 to 20 minutes. Its outer side not going to melt. So what is been done? The protein from the psychrophiles are isolated and added into it. So it can withhold the temperature for a longer time. And we are happy to eat it because it's not going to harm. It's going to help us only. So like that, so many applications are there. Right now, we are not looking to work out it. Okay. So I'm ending here.